Okay. So today we'll be discussing about the histology of pancreas and suprarenal gland. Now, in case of pancreas, okay, if you remember in the last week, I have explained something regarding stroma, parenchyma, then connective tissue capsule, right? So same story, same concept will be followed over here. In case of pancreas also, same things are present. Okay. So if you see the histology of pancreas, first of all, we know that pancreas is both exocrine and endocrine gland. So two parts are there. Whenever you are studying, whenever you are writing about the pancreas, you have to segregate it. Okay, that means the exocrine part and the endocrine part separately. Okay, don't mix it. Now, before starting with the exocrine and the endocrine part, what you have to know is about the connective tissue capsule, okay, about the septa, about the lobes, okay, which is almost similar to the liver. Okay. So, in case of pancreas, if you see, it is also covered by a connective tissue capsule, okay, and that will from that the septa will extend and will divide the gland into lobules. Okay, so in case of pancreas, connective tissue capsule extend inwards as septa and divides the gland into lobules. Same like the liver, which we have discussed earlier. Here also you are getting stroma and parenchyma. Okay. Now here, what is present in case of stroma and what is present in case of parenchyma? In stroma, mainly the connective tissue capsule is there. From connective tissue capsule, septa is extending into the gland. That will be the stroma. Followed by, we are getting the blood vessels, nerves, lymphatics. Okay, those will also come under the supporting part. That is the stroma. Okay. Next, if you want to see the parenchyma of pancreas, as I said, it is divided into two parts. Okay, that is the exocrine part and the endocrine part. Okay. Now, the parenchyma of the pancreas is mainly made up of pancreatic acini, okay, and pancreatic islets. So these are the two structures which are forming the pancreatic parenchyma. What are they? Pancreatic acini, that is for exocrine part, and pancreatic islets for endocrine part. Okay, these are the two parts which are forming the parenchyma of the pancreas. Okay. Now, interlobular connective tissue, which is forming the septa, will project from the capsule into the pancreatic parenchyma, and it will organize the lobules. Okay. Now, in that septa, what are present? First of all, we will be getting the interlobular ducts. Okay. So the septa, which is extending into the substance, in that we are getting the interlobular ducts, blood vessels, nerves, and lamellar corpuscles. Okay. So these are some important structures which are present, okay, along with the septum, and that is extending into the gland. Now, as I said, in case of pancreas, the parenchyma is divisible into two parts. What are those two parts? Exocrine part and the endocrine part. Okay. Now, the exocrine part of the pancreas is made up of compound tubulo-alveolar serous gland. Okay, compound tubulo-alveolar serous gland, the exocrine part. And the bulk of the pancreatic tissue is formed by the exocrine component. Okay, that means exocrine components are more as compared to the endocrine component. Now, if you see the exocrine component, mainly they are made up of acini, serous acini, okay, in which the acinar cells are there. From those acinar cells, the secretions are taking place. That means the enzymes which are secreted. In the exocrine part, what is present? Mainly the enzymes, pancreatic enzyme, okay, and from endocrine part, hormones are secreted. That I hope you should know. In pancreas, two parts are there, exocrine and endocrine part. So the exocrine part which is there, that is made up of pancreatic acini, where acinar cells are there. Okay, from the acinar cells, the secretions which are taking place, these are nothing but the pancreatic enzymes. Okay, and if you see the islets, pancreatic islets, what is present in the pancreatic islets? Particular cells which are producing hormones. Now, the exocrine component is important because it helps in the digestion. Okay. Now, as I said, it is made up of the pancreatic exocrine. It is made up of pancreatic acini, and inside the acini, acinar cells are present. Okay. Next, if you see the endocrine component, it is much more smaller as compared to the exocrine one, and the endocrine part is mainly made up of pancreatic islets. Okay. Now, if you see the appearance of the pancreatic islets, okay, these are in the form of islands of cell dispersed between the pancreatic acini. Okay, so pancreatic acini will be there throughout the gland, okay, and in between them, in some areas, the island of cells will be present. Okay, those are called as islets, pancreatic islets or islets of Langerhans. Okay, now the islet cell produce and secrete hormones. Okay, so the cells of the islets, what is the main function? The main function is they will produce hormones, and those hormones will regulate the glucose, lipid, and protein metabolism of our body. Okay, so as I have mentioned, please note that in case of pancreas, whenever you are writing about the histology, you have to segregate it. Okay, that is the parenchyma is divided into two parts, exocrine part and the endocrine part. Exocrine part में क्या रहता है? What is present in the exocrine part? Pancreatic acini, okay, which is made up of acinar cells, from which the pancreatic enzymes are secreted. What about the endocrine part? It is made up of islets, okay, island of cells. From there, what is produced? Hormones are produced, and those hormones which are produced, okay, what is their function? Their function is they will regulate the glucose, lipid, and protein metabolism of our body. Okay, now we'll start with the pancreatic acini. Now, if you see the pancreatic acini, as I said, these are branched acinar gland surrounded by incompletely lobulated delicate connective tissue. Okay, that means if you see the pancreatic acini, they are branched acinar gland and they will be surrounded by delicate loose connective tissue. If you see the epithelial lining of the acinar cells, what is the epithelial lining? It is simple columnar epithelium. Okay, and if you see the shape of the cell, first of all, simple columnar epithelium, along with that, luminal part is narrow and the basal part is broad. Okay, the nucleus which is present is spherical and that is basally placed. So in case of columnar epithelium, we know that the nucleus which is present that is at the base of the cell. Next, if you do H&E staining, okay, of the pancreatic acini, H&E staining of the pancreatic acini, so the cell shows basal basophilia and apical eosinophilia. Okay, that means same stain, but the appearance in the cell, okay, the stain which is taken by the cell and the apical part and the basal part, it is not same, it is different. Okay, that's why basal basophilia. So like this you can remember, B is basal, B is basophilia. Okay, so the basal part will appear basophilic and apical part will appear eosinophilic. Okay, now why exactly it appears? So the basal basophilia is because of rough endoplasmic reticulum, while apical eosinophilia is because of numerous secretory vesicles, are also called as zymogen, granules, and Golgi apparatus. Okay, because of these particular parts which are present at the basal and the apical part, the staining differences can be seen. Okay, and this is also an important identifying features of the acinar cells which are present inside the acini. Now, these zymogen granules, these are very important because these are the secretory granules which contain digestive enzymes. Okay, so various digestive enzymes are present. For example, protease, amylase, lipases, nuclease. Okay. Now, these zymogen granules, they increase in the size during the fasting. Okay, and from the granules, secretions which are taking place, that process is called as exocytosis. Okay, next, another important type of cell which can be seen under high magnification, that is the centroacinar cells. Centroacinar cells. Now, these are located at the center of the acini. Okay, so if you take one acini, at the center of the acini, there are some cells present. Those are called as centroacinar cells. Now, these particular cells are not the secretory part. That means they are not secreting anything. Rather, they are the part of the ducts. In exocrine pancreas, we are having duct. Okay, exocrine pancreas, ducts are present. That means all the secretion, whatever taking place from the pancreatic acini, how it will be conveyed? It will conveyed by the duct system. Okay, and coming into the duct system. So this centroacinar cell means at the center of the acini, a particular type of cell will be there, and the appearance is different from the acinar cell. Why? Because these are not acinar cells. These are the cells of the duct. Okay, which is invaginated into the acini. Okay. Now, centroacinar cell, if you see the centroacinar cell, shows pale staining cytoplasm with centrally placed flattened nucleus. Okay, and centroacinar cell secrete bicarbonate ion into the pancreatic juice. This is the function of the centroacinar cell. Okay. Now here you can see the diagram. So I was talking about the pancreatic acini. Okay, so this is one acinus where you can see these are the acinar cells. Okay, these are the acinar cells, and at the apical part of the acinar cells, you can see the zymogen granules are present, from which the secretion is taking place. Okay, next you can see this is the duct, intercalated duct. So this duct is invaginating inside the or invaginating into the acini. So though it is invaginating into the acini, so at the center you can see some cells which are there, and the appearance will be different from the acinar cells. Okay. So these particular cells are not part of the acini. These cells are the part of the duct. Okay, and they are present at the center of the acini. That's why they are called as centroacinar cell. Okay, and these things can be seen under microscope in high magnification. Okay. The most important function of the centroacinar cells are, once the secretion is taking place, pancreatic secretion, the exocrine secretion, it will add bicarbonate ion into the pancreatic secretion. That is the important function of the centroacinar cells. Okay. Now this is a schematic diagram. Okay. This is a schematic diagram of the pancreas. Okay, where you can see these are the connective tissue septum. These are the connective tissue septum, and this is one lobule. This is one lobule. Now inside this, you can see these are the pancreatic acini. These are the pancreatic acini. Okay. Now in between the pancreatic acini, you can see these are the pancreatic islet. Okay, these are the pancreatic islets. This is one islet. This is one islet. Okay, this is one islet. So majority is made up of pancreatic acini. Okay, majority. In the islands of cells will be present. Those are called as pancreatic islets. Okay, so here you can see the pancreatic islets are present. Okay. So as we go further, okay, the things will get clear. Next is the duct system. So we have seen the pancreatic acini, we have seen the acinar cells, how the secretions are taking place. Now once the secretion is taking place, it has to be conducted. Now do you remember the ducts of the pancreas which we have discussed last week? I have explained the pancreas in the class, and I have shown you the ducts also. If you remember, main pancreatic duct, accessory pancreatic duct, right? And if you see the main pancreatic duct, it is having herringbone pattern. Okay, those things we have discussed. Now the pancreatic secretion, whatever I am talking about exocrine part, when the secretions are taking place, where it is going? The secretions which are taking place from the pancreas, I am talking about exocrine part, where the enzymes are present. Where it will go? It will go to the second part of the duodenum. How can you forget all those things? पेंक्रियास से जो एंजाइम सीक्रेट होता है कहाँ पे जाएगा? Where it will go? Apply common sense. It will go to the second part of the duodenum. Before draining into the second part of the duodenum, it will bind with the which duct? Common bile duct to form hepatopancreatic ampulla or ampulla of Vater. Several times I have mentioned this in the classroom. And in case of any exocrine gland, ducts are there that you should know. I don't have to explain this thing inside the classroom. कोई भी exocrine gland है, उसका जो secretion होता है, that secretion is carried by the duct. And if it is endocrine gland, directly it is poured into the bloodstream. That much concept should be known by all of you. Now, in case of exocrine pancreas, after the pancreatic acini, the duct systems are there because that duct has to convey the secretion, mainly the pancreatic juice. Now, in exocrine pancreas, as I said, duct systems are there. Now, if you see the duct, first of all, intercalated duct is there, the smallest one. Okay, then interlobular duct, and then main and accessory pancreatic duct. This is the duct system. This is how the pancreatic enzyme or pancreatic juice is conveyed by the duct system. Okay. Now beyond the interlobular duct, okay, it is surrounded by connective tissue septa containing the smooth muscle and the autonomic nerve fibers. Okay. Next, if you see the intercalated duct, now this intercalated duct, these are intralobular duct that begin as centroacinar cells. The centroacinar cells which I have shown you just now. Okay. Now if you see those particular duct, they are simple squamous epithelium. Okay, to very low cuboidal epithelium. So every time you have noticed that first you have to know the duct system from where it is starting and where it is ending, and each and every duct as it is going out, what is the lining epithelium. So here it starts from the simple squamous epithelium, okay, to very low simple cuboidal epithelium. Now intercalated duct, it will open into interlobular duct. Okay, intercalated duct will open into interlobular duct, and that is lined by cuboidal to low columnar epithelium. So as you will go towards the larger duct from the smaller one, okay, epithelial lining starts changing. Okay, that means first it will be similar to the simple squamous cell, then low cuboidal, then cuboidal, and then columnar. Okay, this is the transition which changes as the diameter of the duct increases. Now, the main and the accessory pancreatic ducts, these are lined by simple columnar epithelium. Okay, and with the help of those ducts only, the enzyme is poured or enzyme is conveyed into the second part of the duodenum, which helps in digestion. Now, the exocrine pancreatic secretion. Okay, the exocrine secretion is under control, and how it is controlled? It is controlled by secretin and cholecystokinin hormones as well as the autonomic nerves. Okay, so these are the hormones which are controlling, and along with that the autonomic nervous system that is also controlling the secretion, exocrine secretion. Now this is a beautiful diagram. Now in this diagram you can see the acinar. This is one acinus. Okay, if it is multiple, then it is called as acini. If it is one, then it is called as acinus. Okay, so this is one acinus, right? Can you see the acinar cells? Beautiful. These are the acinar cells. This is one acinar cell. This is one acinar cell. Okay. Now these dot things, whatever you can see, these are the zymogen granules, which are present at the apical part of the cell. From this zymogen granules only, the enzymes are secreted. Now the enzymes which will be secreted, where it will go? It will go to the intercalated duct. As you can see, this is the intercalated duct. Now here you can see the duct is going inside the acinus. Can you see? The duct is going inside the acinus. So this is called invagination of the duct. Okay, and it is reaching up to the center of the duct. It is reaching up to the center of the acinus. As you can see, this is the duct. It is reaching up to the center of the acinus. Okay, so at the center, the duct cells which are present, okay, those are called as centroacinar cells. Okay, now here you can see the pattern. Intercalated duct is there, then interlobular duct is there. Okay, after that you will be getting the main and accessory pancreatic duct. Now in this particular chart, you can see the lining epithelium which is present at the various part of the duct. Okay, so please keep that in mind. In the smaller duct, it is squamous epithelium to low cuboidal. Then in medium sized duct it will be cuboidal, and large sized duct the lining epithelium will be columnar epithelium. This is the transition, and in most of the cases this pattern is followed. Okay. Now along with the cells, acinar cells, okay, another important type of cells are present in the pancreas, and these are called as PSCs. What are these? These are also called as pancreatic stellate cells. Okay, pancreatic stellate cells, very important. Now, what is the importance of the pancreatic stellate cell? Now, these are predominantly present in the pre-acinar, pre-ductal, and pre-vascular spaces of the pancreas. These are the areas where these particular cells are present. Okay, star-shaped appearance. They are having star-shaped appearance. Now, what is their function? So, these stellate cells, the normal tissue architecture is maintained. They help in maintaining the normal tissue architecture of the pancreas. Okay, how? They will synthesize the ECM extracellular matrix protein, matrix degrading enzymes, okay, tissue inhibitors, metalloproteinases, etc. Like this, they are maintaining the tissue architecture. Now, these particular cells can be activated by factors including pro-inflammatory cytokines, oxidant stress, hypoxia, hyperglycemia, increased interstitial pressure. Okay, because of this, the particular cells can be excited or it can be activated. Now, after that, what will happen? This particular stellate cell will secrete various growth factors to promote growth and proliferation of the pancreatic cancer cell, inhibit apoptosis, enhance their invasion ability. Okay, once it is activated, after that, what it will do? It will produce the growth, or it will enhance the growth of the pancreatic cancer cells, inhibit the apoptosis, and enhance the invasion of that particular cancer. Okay, so that's why these particular cells are very important, the pancreatic stellate cell. Now here you can see the pancreatic stellate cells. Okay, so for that different type of stains are required in order to identify. In normal H&E staining it cannot be seen. Okay, here you can see the appearance of the cell. See the appearance of the cell. Okay, these are the pancreatic stellate cells. Next, the pancreatic juice. That means the exocrine part of the pancreas is secreting the pancreatic juice. Okay. Now, in that pancreatic juice, what is the volume of the pancreatic juice? 500 to 800 ml per day. Okay. And if you see the pancreatic juice, what is the pH? It is 8 to 8.3. That means it is alkaline in nature. 8 or 8.3 means it will be alkaline. It will not be acidic, right? If pH is 8, what will be the nature? Acidic or alkaline? If it is 8, it will be acidic. It will be alkaline in nature. That means the pancreatic juice which is secreted by the exocrine pancreas, it is alkaline in nature. Okay. Now in the pancreatic juice, you can see these are the constituents of the pancreatic juice. So see the water content and see the solid. Okay. Now my focus is to show you the enzymes, different types of enzymes which are present in the pancreatic juice. Please keep that in mind. In the pancreatic juice, only enzymes are not present. Along with the enzymes, other compounds are also there. Okay, you can see other compounds are also present. Now see, different types of enzymes are present: proteolytic enzyme, lipolytic enzyme, and amylolytic enzyme. These are the different types of enzymes which are present in the pancreatic juice. Okay. Next, in this particular chart, you can see the enzyme, pancreatic enzyme. Okay. Then activator, how they used to get activated. Then acts on which substance. You know that any enzyme is there, they will be acting on a particular substance. One, it is acting on a substance, you will be getting the end product. Okay. So here, these are the list of enzymes. This is the activator, acts on which particular substance, and this is the end product. Okay, so these are the different types of enzymes which are present in the pancreatic juice. So I will request everyone to go through it, okay, because it will be required in physiology also. Okay, next is the endocrine part. So we have seen about the exocrine part. Next is the endocrine part. Okay, so if you see the endocrine part, only 2% is made up of endocrine part. Remaining 98% is made up of exocrine part. That means the main bulk of the parenchyma is made up of exocrine part. Only 2% is made up of endocrine. Now, in the endocrine part, approximately 1 to 2 million pancreatic islets are there, or also called as islets of Langerhans. Okay, in case of a pancreas, about 1 to 2 million pancreatic islets or islets of Langerhans are present. Now, if you see the appearance, they are dispersed throughout the exocrine component of the pancreas, and mostly they are situated at the tail of the pancreas. Okay, islets, they are present distributed throughout the gland, but the number of islets are more in which part of the pancreas? Tail of the pancreas. Okay. Now, how you will demarcate the islets? This is very important. You have to differentiate the islets from the exocrine part. How will you differentiate it? Okay. So they are demarcated by rest of the parenchyma by delicate sheath of reticular fibers, which is present in that. Next, if you see the pancreatic islets, these are spherical clusters of polygonal endocrine cell. Okay, clusters of cell will place together, and if you see the appearance, they are pale staining. Okay, under H&E staining, if you see the slide of the pancreas, they are pale staining, and they are enveloped intensely staining basophilic pancreatic acini. So around them, intense staining basophilic acini will be there, pancreatic acini, the exocrine part, and in between them you will be getting the cluster of cells which is pale in staining. So this is how it is differentiated. Now the cells which are present in the islets, different types of cells are there. I am coming into that. Now if you see those particular cells which are present in the islets, they are attached to each other by desmosomes and gap junction. Okay, as a result, they are forming cords of the cell, bands or cords of the cells. Okay, the cells which are there, they are connected to each other by desmosomes and gap junction, forming the bands or cords of the cells. Now, the pancreatic islets, as you know, that pancreatic islet is the exocrine part. So here ducts will not be there. Ducts are absent in pancreatic islets. So instead of that, what will be there? Instead of that, you can see it is permeated by fenestrated capillaries. Okay, the capillaries are present. Why? Because the secretion which is coming out will be poured directly into the blood vessel. That's why the fenestrated capillaries are there, which will allow quick entry of pancreatic hormones into the blood. Okay, so just now I have mentioned about the duct system. Okay, so that duct system is associated with the exocrine part. So whenever you are looking at the exocrine pancreas in under high magnification, then you can see the centroacinar cell, and if you trace the centroacinar cell, you will be getting the duct pattern, the ducts, okay, under high magnification. But in case of endocrine one, instead of that duct system, you can see the capillaries, fenestrated capillaries. Okay. Now on H&E staining, the islets of Langerhans cannot be differentiated. This is very important. That means if we use H&E staining, you can't differentiate the cells which are present in the islets. You can see cluster of cells, pale staining, but you can't differentiate the cells. For that, a special type of stain is required, and that is called Malroy-Azan staining. Okay, this is a special type of stain which is required. Okay, and the fixation method is Zenkar formal fixation method. Now, after doing that, okay, three types of cells can be revealed. What are those? Alpha cells, beta cells, and D cells are also called as delta cell. Now, if you see the alpha cell, it will appear red in color, beta cells will appear brownish orange in color, and delta cell will appear blue in color. Okay, not under H&E staining. For that, a special type of stain is applied. When you will apply that particular stain, okay, the name of the stain is Malroy-Azan stain. That also Zenkar fixation, formal fixation. Okay, after that, these colors can be seen. Next, immunohistological staining method is also there. In that method, okay, in addition to alpha, beta, and delta, you can see other types of cells. For example, F cells are also called as PP cells, D1 cell, and enterochromaffin cell. Okay. So these are the other types of cells that can be seen. How? Immunohistochemical staining. Okay, this is a different type of staining. So under this staining, okay, different cells can be identified. Okay, that is PP cell, D1 cells, and enterochromaffin cell. Next, the autonomic neurotransmitters acetylcholine and non-adrenaline affect the islets secretion. That means the control from the islet that is done by this particular neurotransmitter. Now here you can see the list of cells which is present in the endocrine part of the pancreas. Okay, list of cells present in the endocrine part of the pancreas. Okay, so you can see the alpha cells function is it is produce glucagon. Okay, now it is constituting about 15 to 20% of the cell population in case of islet. Next is the beta cells which will produce insulin. Okay, it is producing about 60 to 70% of the cell population. It means the number of beta cells are more as compared to the alpha cells. Next is delta cells or D cells. This produces somatostatin. It constitutes about 5 to 10% of the islet population. Okay. Next, other types of cells which can also be found in the islets. It includes F cells which produces pancreatic polypeptide, D1 cells, vasoactive intestinal polypeptide, EC or also called as enterochromaffin cell. They will secrete serotonin, substance P, etc. Okay, so their numbers are very less. Mostly alpha cell, beta cells and delta cells. They are forming the most of the population of the islets. Okay. Now, if you see the diagram, in this diagram you can see what are these. These are the pancreatic acini. So see, this is one acini, this is one acini, this is one acini. In between the acini, you can see the cluster of cells which are present. Okay, that also it will be pale staining. Okay, and here inside the islet. So this is the islet. Inside the islet, you can see this kind of capillaries will be there. Instead of duct, capillaries will be there. Why I am focusing more into this? Because in the same tissue, there are some areas where you are getting duct, and there are some areas where you are getting capillaries. Okay, so try to understand the difference. So, if you see the acini, this will be associated with the duct. Okay, duct system will be present over there. But if you go to the islets, cluster of cell pale staining, okay, and the cells are in the form of cords. Okay, they are attached to each other with the help of desmosomes gap junction. Here, instead of duct, you are getting the capillaries. See, you are getting the capillaries. Okay. Next, the innervation of the endocrine part is important. Okay, and it is mostly done by the parasympathetic system. Mostly. Okay. Now, fine branches will ramify among the cells to form the plexus around the islet. That means the postganglionic parasympathetic fibers, okay, they will ramify. That means they will redivide into smaller branches, and then they will form a plexus of nerve around the islets. Okay. Now fibers first they will supply the synapse with the acinar cells before innervating the islets. Okay. So cells will be acinar cells will be supplied first, innervated first. After that the islets. Now many fibers enters the islets along with the arterioles. Okay, so the arterioles which are entering the islets, along with that the nerve fibers are also entering. Okay. Next, the parasympathetic ganglia. Okay, I will show you the diagram where the parasympathetic ganglia are also present, lie on the connective tissue within and between the lobules, and these are frequently associated with the islet cells. So together these nerve fibers, the arterioles and the connective tissue which is present, they are forming a structure, and that is called as neuroinsular complex. This is called as neuroinsular complex. Okay. Now both the alpha and the beta cells are involved in these neuroinsular complexes. Okay. Now see this diagram. Okay, this is a 3D diagram where you can see this is the pancreatic islet. Okay, in the pancreatic islet you can see different types of cells are there. Okay, you can see all the types of cells, and you can see they are attached to each other. Okay, with the help of desmosomes and gap junction. Now in between that, what are these? These are nothing but the fenestrated capillaries. You can see these are the fenestrated capillaries. Okay. Now if you go to this particular part, okay, if you go to this particular part, what you can see over here? See, this is the ganglion. So these fibers will be the preganglionic fibers. These are the postganglionic fibers. Okay. So this is particularly the parasympathetic ganglion. In parasympathetic ganglion, first of all, these are the preganglionic fibers which will rely over here, and then you can see the postganglionic fibers are arising. Now these postganglionic fibers will go, and then it will supply, as you can see, these are supplying the cells which are present in the islets. Okay, so like this, they are controlling the secretion of the hormones. Okay, so next, this is the histological diagram of pancreas under low magnification. So what you can see over here, can you see the acini? So see, this entirely, most of the part is made up of acini. In between that, can you see pale staining areas? Here these are the pale staining areas. These are the pale staining areas. Okay. Now these pale staining areas, whatever you can see, these are nothing but the islets. Okay, so under low magnification, that much can be seen. Now if you want to see more, then you have to zoom under 40x. Once you zoom under 40x, then what you can see, this intercalated duct can be seen easily. Next, if you zoom under 40x, okay, the pancreatic islets, whatever it is present around the pancreatic islets, you can see the capillaries also. Okay, so identification of the identification of the islets, okay, under high magnification is quite easy. It's not that difficult. Now see, this is under high magnification. So what is this? This is pancreatic islet, and these are the acini. Okay. Now in the pancreatic islet, you can see different types of cells are there. Now here you can't identify which is alpha cell, which is beta cell. Why? Because it is under H&E staining. In H&E staining, you can't identify. For that, separate stain is there, Melroy-Azan stain. Okay, but you can see this is pale stain, this is dark stain. Like this, you can differentiate. Okay. Next, you can see the Centroacinar cells also, and if the Centroacinar cell is there, if you trace, you can see the duct. Next, inside the endocrine part, that is the inside the islets, you can see the blood capillaries also. Okay. So like this, it can be differentiated easily under high magnification. Okay, so that is all about the pancreas. Okay, so the next topic which we will be discussing, that is the histology of adrenal gland. Anyone, have you seen the adrenal gland in the dissection hall? Yes. Okay. While removing the kidney, I hope you have seen the adrenal gland. Okay. I hope you have studied about the adrenal gland also. Do you know the size and shape of both the adrenal glands are not same? It is different. See, just see the diagram. It is not same. It is different. The right one, just see the shape of the right one. You see the size, shape of the left one. It is different. Okay. I hope you have studied. If not, please study. Those things we'll be studying in the gross anatomy part. Okay. So we'll start with the histology. Now, here also same story is repeated, same like the pancreas. Okay, as you can see, the gland is also surrounded by connective tissue capsule. Okay, and it is sending septa inside the gland. So that is the stroma or the supporting part. And along with the septa, blood vessels will be nerves that will be going inside the gland. Okay. Next, if you see the parenchyma. If you see the parenchyma of the adrenal gland or suprarenal gland, it is divisible into two parts. The outer part is the cortex, larger part. Inner part is called as medulla, smaller part. Okay. In case of parenchyma of suprarenal gland, it is divisible into two parts. Outer, larger part is called cortex. Inner, smaller part is called as medulla. Now, parenchyma of the adrenal gland is divisible into two zones. Superficial is cortex 90%, and deep part is the medulla, only 10%. Okay. Now, what is present in the cortex and what is present in the medulla? Now, this adrenal gland is completely an endocrine gland. Different type of hormones are secreted from the endocrine, this adrenal gland. If you see the cells of the suprarenal cortex, it is made up of cells arranged in the form of cords, and sinusoids are present in between the cords. Okay, so the cells which are present in the cortex, they are in the form of cords, but arrangement is different. Okay, and in between the cords, what is present? Sinusoids are present. Now, on the basis of the arrangement of the cells, in the cortex, okay, it is divided into 3 layers. Okay, it is divided into 3 layers. What are they? Zona glomerulosa, zona fasciculata, and zona reticularis. So as I said, cortex is larger. Approximately 90% of the gland is made up of cortex. Only 10% is made up of medulla. If you see the cells of the cortex, they are in the form of cords, and in between the cords, sinusoids are present. Okay. Next, depending on the arrangement of the cell in the entire cortex, okay, it is divisible into 3 parts. What are they? The outermost part is the zona glomerulosa, where 15% is occupied. Then middle part is called as zona fasciculata. 80% is occupied by this. That means this is the largest part of the cortex. And inner part is called as zona reticularis. Only 5% of the cell population are present. Okay. Now the cells of the adrenal cortex are steroid secreting cells. So they do not secrete. Okay, whenever it is required, means they will not store the hormones inside the cell. Whenever it is required, immediately they will produce the hormones. Okay, this is the speciality of the cells of the cortex in case of adrenal gland. Now, if you see the diagram, so this is the cortex, okay, this is the capsule, this entire part is the cortex, and only this part is the medulla. Okay, that means 90% of the gland is made up of cortex, only 10% is made up of medulla. Okay, so in the cortex, as I said, 3 different zones are there. Okay, the outermost is the zona glomerulosa, then zona fasciculata, and then zona reticularis. Zona means zones, so 3 different zones are there. Okay, now in those zones, cells are present and they are in the form of cords, but the arrangement of the cells are different in these 3 different zones. Okay, based on that you have to identify the zone under microscope. That's why you have to know about that. Followed by the hormones which are secreted by these particular zones. Okay. So first we will see the outermost zone, that is zona glomerulosa. So this zona glomerulosa is the outermost layer. Okay, outermost zone. Approximately outer 1/5 of the cortex is made up of this zona glomerulosa. Now if you see the cells, they are closely packed and they are in the form of inverted U-shaped cords. They are in the form of inverted U-shaped cords. Okay. And along with that, they will be surrounded by capillaries. Now if you see it under light microscope, the cells of the zona glomerulosa are seen to be small, polyhedral or columnar. Okay, so in case of zona glomerulosa, what is the shape of the cell? It is polyhedral or columnar with basophilic cytoplasm and deeply staining nuclei. Now what is the function? They secrete mineralocorticoid hormone, that is the aldosterone and deoxycorticosterone. Okay. Now what is the function? Now this maintains the electrolyte and water balance of the body. Okay, this is the function of the zona glomerulosa. Next, the middle layer or the largest layer or largest zone is the zona fasciculata. Now in the zona fasciculata, first of all, it is the middle layer. Okay, 3/5 of the cortex is occupied by the zona fasciculata. If you see the cells, okay, they are present in the form of column, straight columns, that also 2 cell thick. Suppose one column is there, another column is there, another column. Okay, like these straight columns are there, and in between the columns of the cells, the capillaries are present. Now under light microscope, okay, the zona fasciculata are seen to be large polyhedral with basophilic cytoplasm and vesicular nuclei. Now in this particular cell, okay, they are rich in lipid. Okay, they are rich in lipid, and during the processing, these lipid droplets which are present, they are washed away and they appear empty or vacuolated. That's why they are also called as spongiocytes. And this cell contain considerable amount of vitamin C. Now what is the function? The function is, if you see the function is they secrete glucocorticoid, that is cortisol, cortisone and cortisol. What is the function? They appear to decrease the antibody response and have anti-inflammatory effect. Okay. So this is the main function of the hormones which are secreted by zona fasciculata. Okay. Next is the zona reticularis, the innermost layer. Now zona reticularis, 5% of the cortex is taken by this zona reticularis. Okay. Now here how the cells are arranged. Here the cells are arranged in the form of cords, but it appears as network-like appearance. Okay. As you can see, zona reticulate cells are arranged in anastomosing cords, okay, forming reticulum or network-like structure. Now the cords are, here also cords are separated by capillaries. So if you have noticed that in each and every case, okay, the arrangement of the cells are different. Previously in the first case we have seen inverted U-shaped, okay. Next in the middle part it is straight column, okay, and here it is network-like. But in each and every case, in between the cords of the cells, what is present? Capillaries are present, okay. And the hormones which are secreted by them, directly it will be poured into the blood vessel. Now the cells of the reticularis, these are smaller as compared to the zona fasciculata, okay. They darkly staining nuclei, well developed, smooth ER is present, and along with that few rough ER is also present, okay. Abundant yellow brown lipofuscin pigments are also present inside the cell. Okay. Now cells of the reticularis, okay, secrete weak androgen and small quantity of glucocorticoids. Okay, these are the function of zona reticularis. Now in this diagram, so this is the diagram of schematic diagram as well as histological diagram under low magnification. Okay, where you can see different zones. Okay, so first you can see the capsule. Okay, then here you can see the zona glomerulosa. Okay, see this is the zona glomerulosa. Then this is the zona fasciculata, the middle part, and this is the zona reticularis. Okay, and this particular part is the medulla. So medulla I have not discussed, we'll be discussing about that. Now here you can see the arrangement of the cell. Now in order to see the arrangement of the cell, further high magnification is required to see how the arrangement of the cells are present. Okay, but I hope you can see along with the cells, the capillaries are also present. I hope you can see the capillaries also. Okay. Next is the suprarenal medulla. So suprarenal medulla, functionally, embryologically, it is totally different from the cortex. Okay. In case of suprarenal medulla, what is present? Sympathetic ganglion with postganglionic sympathetic cells are present. Sympathetic ganglion, postganglionic sympathetic cells are there, but axon and dendrites are absent. Next, it is having two important types of cells, that is the chromaffin cells and the sympathetic ganglion cells. These are the two types of cells which are present in the medulla. Okay, now cells, particularly in the medulla, are modified postganglionic neurons that have secretory function. Now, this particular chromaffin cells, if you see, they are polyhedral and have basophilic cytoplasm. Now, the cell groups or columns are separated by white sinusoids. So, here also white sinusoids are also present. Okay. Now, these chromaffin cells, they take potassium dichromate. It is a special type of stain. That's why they are also called as chromaffin cells. Okay. Based on the staining, the names are given. Now, if you see the function, so chromaffin cells secrete hormones, that is noradrenaline and adrenaline into the blood. Okay. Now, the secretion takes place mainly at the time of stress and result in widespread effect, those of the stimulation of sympathetic nervous system. Okay, adrenaline and noradrenaline. And this secretion of the suprarenal medulla is mainly controlled by the splanchnic nerves, that means the sympathetic division. Okay. Further, here you can see the diagram. In this diagram, 3 zones can be seen. First of all, this is the capsule. Then this is under high magnification, where you can see the zona glomerulosa. In inverted U-shaped clusters of cells can be seen. Okay. Next, you can see the zona fasciculata, this part and this part, where the columns of cells are there, and in between that, the capillaries can be seen. Okay. And here you can see the zona reticularis, where networks of cords of cells are present, separated by sinusoids. Okay. And next, this is the medulla. In medulla, as I say, 2 types of cells are there. Secretory cells, as you can see, these are the secretory cells, and then sympathetic neurons are also there. So sympathetic cells are larger as compared to the secretory cells. This is how you can easily identify under H&E staging. Okay. Now lastly, this is the list, or you can say this is the chart where different parts of the gland and different hormones and functions of that. Okay. So you can see the adrenal cortex, zona glomerulosa, outermost layer, the middle layer fasciculata, innermost layer is the reticularis and adrenal medulla. Okay. And what are the hormones secreted? Already we have discussed, but this is the gist. Okay. Means if you want to revise it quickly, this will help you to revise the entire suprarenal gland.